Sickle cell disease is a genetic disorder caused by mutations in the beta-globin gene that leads to faulty hemoglobin protein called hemoglobin S. Hemoglobin S changes flexible red blood cells into rigid sickle-shaped cells. These sickle cells can block blood flow and result in pain and organ damage. Researchers are testing ways to use genome editing technologies such as CRISPR-Cas9 for sickle cell disease. One way is to turn on the gene for fetal hemoglobin, which can help reverse sickle cell disease. Several months after birth, a molecular switch inside our bodies shuts off the gene for fetal hemoglobin and turns on the adult hemoglobin gene. In sickle cell disease, only the adult hemoglobin is mutated and makes hemoglobin S. To turn fetal hemoglobin back on, doctors first need to collect some of the special stem cells from your blood or bone marrow. These stem cells make new red blood cells. The cell's DNA can then be edited in a lab. Scientists usually use a vector to get all the genome editing ingredients inside the cells. One ingredient is a scissors-like protein. Another is a guide molecule that brings the scissors to the spot on your DNA that needs to be changed. The scissors cut the DNA, leaving a gap. In this example, the guide molecule seeks out the molecular switch gene that shuts fetal hemoglobin off. The scissors cut the DNA near the gene. The repair process disables the gene. Without the molecular switch, the cells make more fetal hemoglobin and less adult hemoglobin. These edited stem cells are returned to the body through an IV. The edited cells make healthy red blood cells.